My name is Susie Huang, and I am a neuroradiologist at Massachusetts General Hospital and an associate professor of radiology at Harvard Medical School. Today, I'll be discussing quantitative MRI in clinical practice, progress and challenges. I'll specifically discuss the translation of quantitative MRI as it applies to white matter diseases. So what is white matter in the human brain composed of? It consists of tightly packed myelinated and non-myelinated axons as well as glial cells. And therefore, pathology affecting the white matter primarily involves these components, that is, demyelination, axonal degeneration and loss, and gliosis. So what do we measure with MRI? Within an MRI voxel, which has a spatial resolution of about a millimeter or at most submillimeter, you can see that there's hundreds to millions of axons and nearly that many glial cells as well. So MRI, in fact, only indirectly captures the effects of these various tissue components on the average signal. And it turns out that the range of available contrast mechanisms with MRI enable us to capture different aspects of the white matter signal composition. Quantitative MRI promises to reveal key features of white matter pathology, and in my talk today I will use as an example multiple sclerosis, which is the most common demyelinating disease of the central nervous system. So the hypothesis and premise of using quantitative MRI is that we can better quantify the extent, type, and spatial distribution and evolution over time of tissue damage in patients with multiple sclerosis using quantitative MRI. And my talk today will focus on various contrast mechanisms through qMRI that enable us to capture different features of MS pathology, including pathology in the normal appearing white matter and gray matter, the heterogeneity and evolution of lesions, brain barrier disruption to some degree, and also this really intriguing prospect of remyelination. I'll begin by discussing the role of T1 relaxometry in assessing disease status and progression. So on the panel here on the left, I'm showing representative clinical contrasts that we typically use to detect lesions and tissue loss over time in diagnostic imaging. So FLAIR, 2D FLAIR, and the 3D space FLAIR images are very helpful for identifying white matter lesions. Cortical volume can be assessed very well through T1 weighted imaging, such as the MP-RAGE sequence. And more recently, this double inversion recovery sequence allows us to highlight both lesions in the white matter as well as in the gray matter. So it turns out that there are sequences that have been developed that allow us to actually map out the T1 weighted signal evolution, here exemplified through the MP2-RAGE sequence. MP2-RAGE is a 3D MP-RAGE derivative that provides homogeneous T1 weighted images and simultaneous T1 maps that can be calculated from the series of images. And it turns out that T1 relaxometry mapping studies have been used to generate whole brain assessments of multiple sclerosis disease impact and progression. So these studies in the past have found that the global T1 relaxometry changes actually increase with disease progression, predominantly in the later disease stages, and also correlates with brain atrophy. In addition, there's this prospect that being able to quantify T1 throughout the whole brain may provide a personalized approach in which we could assess the heterogeneity of damage in focal lesions, as well as the extent of diffuse pathology by basically quantifying the changes in T1 relaxation times compared to a normal distribution of T1 relaxation times in healthy individuals. So in this paper here, done by Cristina Granciera's group in Lausanne, MS lesions were basically visualized, and T1 relaxation times were quantified within the MS lesions, which were then used to calculate a T1 z-score. And these z-score maps show that the lesions often appear to be larger than the manually segmented lesions, probably due to areas of increased T1 relaxation time surrounding the MS lesions that were not perceived as lesions per se by expert readers. There are a host of myelin-sensitive metrics that can be derived using quantitative MRI, and these metrics are essential for investigating MS pathophysiology, as it can help us probe lesion evolution and predict outcome, including the effect of therapy. So there's two main quantitative MRI methods for characterizing myelin content, namely quantitative magnetization transfer, or MT for short, and multi-exponential T2 relaxometry. So quantitative MT essentially quantifies the fraction of protons bound to macromolecules like myelin, and it does so by applying selective saturation of protons bound to macromolecules such as myelin, thereby reducing their longitudinal magnetization and rendering it possible to create these magnetization saturation images, or alternatively to be displayed as the ratio of images with magnetization transfer saturation on and off. So these magnetization transfer ratio images, MTR, these are calculated by taking an image with saturation on versus off, and MTSat, is basically an alternative approach that allows us to correct to some degree for the saturation pulses, which can to some degree affect the T1 relaxation times. So in terms of alternative methods to map out myelin, there's also the myelin water fraction that can be derived from multi-exponential T2 relaxometry. So it turns out that T2 relaxometry allows us to be sensitive to different water pools, including myelin water. And this can be basically explored within the central nervous system, primarily in white matter. So both MTR, MTSat, which I haven't shown here, as well as short T2 mapping using multi-exponential T2 relaxometry have been all used to study myelin content and characterize lesions in multiple sclerosis. One of the most important aspects of this work in the past has been actually to validate that these various MRI metrics are sensitive to myelin and specific enough that they may be able to, say, detect areas of demyelination versus remyelination. So focusing first on magnetization transfer, the MTR has been validated by postmortem studies and showed high correlations with myelin when lesions and normal appearing white matter were considered together. Similarly, with using multi-exponential T2 relaxometry, myelin water fraction shows strong correlations with myelin staining in both lesions and the normal appearing white matter, as you can see here on this histological stain, the Luxol fast blue stain, which is binds to myelin. So in the plots below, you can see that there's a strong correlation between myelin water fraction as quantified by multi-exponential T2 relaxometry and LFB optical density, essentially reflecting the myelin content within the tissues, both within averages as well as normal appearing white matter. So using these techniques, which have been supported by histopathological validation, many groups have gone on to assess the prognostic value of MTR and myelin water fraction obtained using these quantitative techniques. The feasibility of measuring dynamic changes in MTR in acute gadolinium enhancing lesions has been demonstrated, as shown in this study here on the left. With acute lesion formation, the average lesional MTR has been shown to initially decrease and then increase. These changes are partially related to edema, but they're also thought to reflect demyelination followed by partial remyelination. The differential effects on MTR recovery in acute lesions have been demonstrated as well for specific therapies, suggesting variations in remyelination capacity. Similarly, myelin water fraction has been studied as a surrogate of myelin content, and has been shown to increase over 6 months following the appearance of enhancing lesions, as would be expected in repairing tissue. And so this brings me to this table here summarizes some of the advantages and limitations to using magnetization transfer imaging and myelin water imaging in clinical trials and clinical practice. Notably, MT imaging is very practical to use. It's implementable on most commercial scanners. It has high precision, but calibration may be required across and within platforms, which is further discussed in Katie Keenan's talk earlier in the symposium. And myelin water imaging has very good intra and intersite reliability and is reproducible, but it has required historically long acquisition times, and there is active research ongoing to try to shorten up those acquisition times. Moving on to other types of quantitative MRI techniques, it turns out that one of the most elusive quantities to measure in multiple sclerosis is axonal damage. So axonal damage is thought to be the substrate of progressive clinical disability, and imaging correlates for axonal damage using MRI have been limited. And these include measures from diffusion tensor imaging, MR spectroscopy. So there's a real need for an improved biomarker of the underlying axonal integrity as a way to assess the therapeutic effect of various neuroprotective drugs that are coming online. So among the markers that have been studied extensively for their sensitivity to axonal integrity include diffusion tensor imaging. Diffusion tensor imaging is sensitive to diffusion both along as well as perpendicular to the axon. It's been very well studied in multiple sclerosis. So quantities like fractional anisotropy are known to be decreased in MS lesions. Mean diffusivity is generally increased. And then axial diffusivity is thought to relate to axonal integrity, whereas the radial diffusivity is thought to be increased in the setting of demyelination. However, various histologic imaging correlation studies have shown that these measures are not entirely specific to axonal integrity. For example, radial diffusivity reflects not only demyelination, but also a component of axonal loss. And this has led to the search for more specific markers of axonal integrity and microstructure using advanced diffusion imaging models and acquisitions. Based on parametric representations of the diffusion MRI signal, a multitude of tissue microstructural parameters can be inferred, such as the relative volume fraction, say, of water within axons in an extraaxonal space, the compartment size of axons, such as average axon diameter, and their orientation distribution. So one advanced diffusion model that has gained traction in the last decade or so is NADI. NADI stands for Neurite Orientation Dispersion and Density Imaging. And this was designed as a clinically feasible multi-B-value protocol that can inform on relative axonal volume fraction independent of fiber crossings and dispersions. So NADI employs a 3-compartment model consisting of neurites, which are essentially axons or dendrites, the extraneurite space, and isotropic diffusion within, say, the cerebrospinal fluid. So the metrics that we can pull out from NADI quantitatively include the intracellular volume fraction, orientation dispersion index, and then the fraction of isotropic water. So NADI has been used in a number of studies in multiple sclerosis, and in general, these studies show that there is overall decrease in the neurite density index within focal lesions. This was shown in the brain by the group at University College London, also corroborated in spinal cord studies. And then in a larger population, the study was published in Brain that basically showed and confirmed that there was an overall decrease in intracellular volume fraction within lesions and increase in the isotropic volume fraction. So as a quick summary, in terms of proposed imaging techniques and their role in assessing particularly neuroprotection, DTI is advantageous in the sense that it's very reproducible and has high sensitivity to changes in the tissue, but it's quite limited in its specificity. Whereas advanced diffusion imaging metrics like NADI have gained quite a bit of interest in the last few years for their ability to quantitatively assess axonal metrics, but further validation studies are needed, and in particularly when we want to consider say, small diameter axons, even quantify axonal size, we may need to use higher gradient systems, which are not very widely available at this time. Finally, in terms of other tissue contrasts that can inform about lesion status and particularly the presence of chronic inflammation. So quantitative susceptibility mapping, or QSM, reveals the magnetic properties and has sensitivity to iron and myelin in white matter. So the longitudinal measurements of QSM, the consensus of various studies is that the susceptibility of lesions increases in the initial phase and then essentially normalizes in chronic lesions. And this is thought to reflect the kinetics of iron deposition to some degree in MS pathophysiology, and provides basically a connection between prior histochemical and MR imaging observations of increased iron within lesions as a way to track disease activity over time. So it's known that both myelin loss as well as increased iron content contribute to increased magnetic susceptibility in MS lesions, and this has to some degree created debates about how to interpret increased magnetic susceptibility. A more recent finding has been that some chronic lesions actually exhibit a paramagnetic rim on susceptibility weighted imaging, particularly on the phase and QSM images. But also you can see this on T2 star weighted images and maps. And this persistent phase rim after enhancement resolves. So this was a study that was done at 7 Tesla, showing that despite resolution of enhancement within this lesion, there was actually the development of a persistent phase rim, this dark rim around the lesion. And this is thought to correspond to chronic inflammation at the boundaries of the lesion, specifically in reflecting iron content taken up by microglia at the edges of the lesion. This paper by Martina Absinta et al. Basically showed that the persistent phase rim predicted poor outcome in new MS lesions. And so this has led to a very promising way of potentially better understanding and characterizing the heterogeneity of MS lesions and predicting outcome based on the prevalence of such lesions. So in theory, quantitative MRI techniques that measure accurately and reproducibly a biologically specific signal correlating with clinical outcomes would be an ideal candidate for clinical application. However, there are several development steps that are still needed for robust clinical use, and these include the clinical availability of the acquisition methods and tools to reconstruct these parametric maps, methods to extract quantitative information from the parametric maps, and normative values, including cutoffs for normal and pathologic tissue. These sort of quantitative maps also have to be considered in the context of existing diagnostic and prognostic criteria that are used clinically these days. So in summary, in this context, I think quantitative T1, to some degree the myelin imaging, appear to be a little bit more mature and technically advanced and ready for use in studies for using quantitative MRI, whereas the QSM and diffusion-based methods do provide information about microstructure, but may warrant further validation as well as technology development for them to be used in a multi-center setting. So with that, I want to thank you for all your attention, as well as members of my group for contributing to our studies looking at various quantitative MRI techniques in multiple sclerosis. Thank you.